So today I will present some neuroimaging data showing that the hippocampus, which is one of our brain structure, may support binding of visual information across gaze fixations when we build our memory. So my research area is memory and study visual memory. As you may know, in a typical visual memory task, you may be given this kind of visual stimuli to memorize. And very likely you will look at a few locations like this. And those locations actually are important because you make fixations. We know fixation is important. They provide high resolution visual information for us so that we can build a better visual representation for the world. So actually, psychologists have found that the fixations to images predict the subsequent memory of those viewed images, independent of those total viewing time. So the fixations, they are important. At the brain level, the hippocampus is also important for binding information to form memory. So now the question comes, should the visual information sampled from the individual gaze fixations are the basic unit of the information that our memory system works on? If that's the case, we may need to develop a new memory research paradigm to examine the memory processing at a better temporal and spatial resolution. For example, focus on fixations. This is important considering the current mainstream memory research paradigm mainly focus on the individual stimuli, the whole picture, like the one I showed you, as the unit of the memory task. So to do that, we conduct two neuroimaging studies to investigate whether the hippocampal activity related to memory can reflect the accumulation of information across fixations. So for example, if I give you this picture to memorize, some of you make more fixations and some of you may make fewer fixations. The question is whether your hippocampus will show stronger activity when you make more fixations compared to when you make fewer fixations. So to do that, I combined the eye tracking with neuroimaging fMRI. So I can measure people's neural activity and their eye movement at the same time. When they do a memory task, they memorize different pictures. In the first study, we use a natural viewing task to investigate this question. We give them many face pictures, let them to process those pictures, then look at whether more fixations direct to the faces will lead to stronger hippocampal activity. And here's the data. You can see this is our human brain. And those warmer color means they make more fixations. Those regions will show stronger activity. And here, this is the hippocampus. So this is a long structure. And this is another section view. So we found bilaterally, here you can understand the beta values is like a regression coefficient, like the prediction. So bilateral hippocampus showed stronger activation if participants direct more fixation to those faces. So this provides preliminary support for our hypothesis, but the data mainly is correlational because we didn't manipulate anything. It's important to experimentally manipulate their eye movement so that we can examine the cause effect, how the fixation can affect hippocampal activity. So that's what I did in the second experiment. I asked participants if they see a green cross, then they should freely explore and memorize this picture. But when they see a red cross, they have to fix on a specific location. At the same time, I gave the same amount of time for them to do this task. And this creates two experimental conditions. One is the fixed viewing, the other is the free viewing condition. Then I can compare their brain activity in these two conditions. And here's the brain picture. This is the surface view. This is like our brain cortex surface, and this is the section view. You see those warmer color regions, they show stronger activation in the free viewing versus the fixed viewing condition. And here, this is the hippocampus. You can see this is the lung structure. So these regions include the hippocampus. Again, this here we provide experimental data showing that the gaze fixations are important for memory processing. So because my interest is in memory, I also analyzed the brain pattern that support memory. You see here, we call it subsequent memory. So these warmer color shows, so these regions will show stronger activation if we memorize some stimulus better. Okay, and this is the pattern inside the hippocampus. You see, this is the brain, this inside the hippocampus. And this brain pattern is the fixation visual exploration effect, which means this region will show stronger activation if we deploy more fixations to the stimuli. And again, here's the hippocampus. So this kind of analysis in the field called brain activation pattern similarity. So we want to see whether the brain activation pattern will be similar between this subsequent memory effect and the visual exploration. And here's the data. You can see indeed there is similarity between the brain activation supporting subsequent memory and the brain activation supporting this visual exploration. However, this similarity only exists when we can freely explore the picture, those visual stimuli. It will be gone in the fixed viewing condition. So that means the visual sampling, visual exploration indexed by those gaze fixations and the memory processing, they are closely related at the brain level. So taken together, we think these results provide empirical support for the idea that hippocampus may bind information across gaze fixations at real time, right? Where we see different things, we'll bind the information to form memory. Therefore, we should consider individual gaze fixations instead of individual stimuli as the basic unit of information for our memory system. We should develop a new paradigm. So with that, I want to thank my collaborators. Thank you for your attention. That's it.